So today I will be talking to you about my research, which is focused on the potential use of genipin, which is basically a compound derived from the plant Gardenia jasminoides. And we are hoping to explore the use of this compound for the management and treatment of eye diseases. So I will start with a brief introduction about the eye in general. So when the light enters the eye through the cornea, which is the clear, transparent layer that covers the front of the eye, the light passes through the pupil, and then the light hits the lens, and then focuses the light onto the retina at the back of the eye, and then transmits the signal to the brain. So all the parts of the eye are equally important and necessary for healthy vision. However, my work has been focused on the cornea, so today I will be mainly talking about the cornea. Just to give you a slight introduction of about the structure of the cornea. So here we can see this is a histological section of the cornea. It is mainly composed of 5 layers. Briefly, the outermost layer is the corneal epithelium. So this layer provides mechanical protection and also protects the cornea from the invasion of pathogenic microbes. We have the Bowman's layer, which is the acellular layer, also providing structural integrity to the tissue. And then we have the corneal stroma, which, as you can see, is composed, is basically taking part nearly 90% of the tissue structure. And at last, we have the Descemet's membrane and the endothelium layer, which basically they are very important in providing nutrients and maintaining tissue hydration. One question, can you guys see the mouse when I move it?
No.
Oh, okay.
Maybe. No, actually, yes, sorry. Yeah, we can.
Okay, because, okay, thank you. So as I mentioned, the corneal stroma is probably one of the most important layers of the cornea, and this is because the structure of the stroma plays a critical role in the maintenance of corneal transparency. So here we can see these two pictures. They are transmission electron micrographs of the corneal stroma. So the corneal stroma is mainly composed of collagen fibers that they are organized into ribbons or lamellae. These fibers, they are orthogonally arranged and they form like a basket weave. In addition to the collagen, we have the proteoglycans, which they are proteins covalently attached to one or more glycosaminoglycan side chains. So these proteoglycans, as you can see, they are very important in regulating the interfibrillar space, so the space between the collagen. So in my group, in the structural biophysics group, we have been extensively studying the structure of the corneal stroma. This is very important also to understand how this is maintained, how it is laid down, and this has potential applications for bioengineering a cornea in the future, and basically the corneal stroma. And here I am showing you a 3D reconstruction of the corneal stroma. This was made by members of my group, of the structural biophysics group. We can see the collagen in blue and the proteoglycans in yellow. And I hope that you can appreciate that it is very important. They are uniform arrangements, so you can see that the spacing between the collagen, the diameter of the fibers, they are very important. So these are very important in order to allow light refraction in the eye and to maintain the corneal transparency. In case someone is interested on the physical aspects, I would like to direct you to this website. This website was done again by the members of my group in Cardiff. And they have really brilliant illustrations of how the light enters the cornea, how light refraction is achieved, and why this structure is very important. Did you ever take a moment to appreciate that we can perceive our environment, whether this is in the air or under the water? And of course, under the water, our vision is blurry. but nevertheless we can see. So the reason for this is that the corneal stroma, and essentially the corneal shape and curvature of vertebrates' eyes, has massively evolved over the years. So the cornea has been, through these evolutionary changes, the cornea has been transformed from being just a cover to being a refractive lens and essentially a camera type eye. And due to the time, I don't have time to go in details about these changes, but if you would like to know more how the corneal has evolved amongst the species and through evolution, I would like to guide you to this review. In case you are not able to get access, please feel free to email me and I will give you a copy. So, as I mentioned, the corneal structure and shape, it is very important for its proper function, and any changes in its structure, they often underlie eye diseases that they affect vision. A fine example is keratoconus. So as you can see here, keratoconus is a progressive eye disease. And as you can see, it is characterized by abnormal bulging of the cornea into a cone-like shape and thinning of the cornea. It often begins in adolescence and adulthood. And in the UK, it occurs 1 in 2,000 people. To be honest, the pathophysiology of this disease has not yet fully understood, but among the most important causes is eye rubbing, allergies, and we also have the genetic component, so it can be inherited. So affected individuals, they are sensitive to light, and they have poor vision. So here you can see how affected individuals, how they see, depending on the stage of the disease. So the current treatment for keratoconus is the corneal cross-linking procedure using riboflavin and UV light radiation. So this is the only keratoconus treatment proven to slow or stop the progression of the disease. So basically, this treatment strengthens the cornea by cross-linking the collagen fibers through the application of riboflavin drops and exposure to UV light. Although this is the gold standard treatment, this procedure, it is not recommended for advanced cases and specifically for cases with very thin cornea. And this is because of associated risks with the use of UV light. Of course, scientists around the world, they have been trying to modify this procedure, to adjust it in order to make it accessible for patients with very advanced cases. And one of the research focuses is to use alternative cross-linking agents that ideally avoid the use of UV radiation. So one of these is genipin. So as I mentioned, it's a natural extract derived from the plant Gardenia, and owing to its excellent biocompatibility and low toxicity, it has established captivating potential in different research areas, including biomedicine and bioengineering. So for example, if you can see the graph here, these are papers, number of papers published by year associated with genipin. So I hope you can appreciate that this is an emerging field, especially for the last years. So the application of genipin in the field of ophthalmology is rarely found. However, it has been reported the successful application of genipin to strengthen the biomechanical properties of the sclera of the eye in order to prevent myopia, to prevent myopic progression, basically. With respect to the use of genipin to cross-link the cornea, Dr. Avila and Navia, they have been the first to report the successful application of genipin for the cornea, and they have reported that genipin can actually provide a corneal stiffening effect similar to the UV, to the conventional approach. However, it is very important to mention that with the genipin cross-linking, we don't need the radiation. So it is very important. So these authors, they have done serious investigations here. For example, I hope you can see my mouse. We have some corresponding corneas. We have the control, which is basically the untreated cornea. And then we have the genipin crosslink cornea. And they put these corneas treated with collagenase. Collagenase is basically one enzyme that can break down collagen. And I hope you can appreciate that with the time, the genipin crosslink cornea is more resistant to this treatment. Also here, you can see the changes in the area between the control and the genipin. And you can also see that the treated cornea is more biomechanically stronger, it's basically stronger. So following these promising results, Dr. Avila and his colleagues, they carried out further investigations, some of them in rabbits, without showing any adverse effects. And they also investigated the toxicity of genipin to the corneal endothelial cells. So this is the cells of the innermost layer of the cornea. So basically, following treatment with different cross-linking agents, they stain these cells with alizarin red. This staining shows the damaged cells. So you can see that genipin has low toxicity compared to other cross-linking agents. And again, Dr. Avila and colleagues, they show that genipin cross-linking can potentially affect the keratometry. So basically, keratometry, this is the corneal curvature, and it can return the cornea back to relatively physiological levels. So in collaboration with Dr. Avila, we are trying now to optimize the cross-linking protocol of genipin. And also in collaboration with Dr. Pedro Castro, we are trying to better understand the photochemical events associated with this natural cross-linker. So the success of collagen cross-linking has fueled the search for alternative applications for this treatment in the field of ophthalmology. So although in early stages, some studies have proposed that this approach can be used for the treatment of infectious keratitis. So infectious keratitis is basically a corneal infection caused by microbes, by bacteria or by fungi. So they term this approach as photoactivated chromophore for keratitis corneal cross-linking. It is very important to find novel therapeutic treatments for infectious keratitis, and this is because it is the second leading cause of corneal blindness, with approximately 10 million people suffering worldwide. And although corneal transplantation still remains an important treatment for these diseases, limited tissue availability, the lack of trained corneal surgeons, and also legal, ethical, religious, and cultural barriers, they make this approach not very easy to be applied. So in order to further emphasize the need for new antimicrobial drugs and treatments, according to the World Health Organization, it is expected that by 2050, more people will die due to antibiotic resistance than to cancer and diabetes combined. So the quest to develop new antimicrobial therapies in combination with the promising data supporting that corneal cross-linking could kill bacteria, irrespective of their antibiotic resistance, and in combination also with the therapeutic potential of genipin that has been proposed by several studies. We went to formulate the research question of whether could genipin cross-linking also be used for the treatment of infectious keratitis. So the first step in answering this question was to first investigate whether genipin has any antimicrobial activity. And we started by first investigating its antibacterial activity against Staphylococcus aureus. This is one of the most causative agents for corneal keratitis. And we ran a simple and relatively quick test, and this was the agar diffusion test. So you culture your bacteria, and then you place some discs, you can see here, with different concentrations of the drug you want to test, in our case, genipin. And then you allow the bacteria to grow, and then you go back after 24 hours usually, and you check whether there have been any areas that they haven't had any bacterial growth. So I hope you can see that in these two concentrations, there is some area with no bacterial growth. So we then went to elucidate the exact action of genipin and to determine whether genipin can just stop bacterial growth, or whether it can also kill the bacteria. And very briefly, we did more tests, and basically we incubated the bacteria with a whole range now of the drug of different concentrations. We also had our control, so we had the bacteria growing alone. Also, we use a standard antimicrobial against aureus that we know that it is killing the bacteria. And then you treat them, you allow the bacteria to grow, and basically then you plate them in agar plates, and then you see whether the drug had any effect on the bacteria. So, for example, these two, there are two different concentrations of the drug. You can see that here, at this concentration, there was no bacterial growth, so it has killed the bacteria. But in this one, the concentration was not high enough to prevent bacterial growth. And we also have our controls. So following these results, and after we determined the minimum inhibitory and bactericidal concentration, we wanted to establish an ex vivo corneal infection model in order to be able to test this further. So in our experiments, we use cadaver porcine eyes. We established the corneal infection model. This entailed various challenges. One of them was to make sure that the corneas were infected only with the bacteria we wanted to introduce, and not with other environmental bacteria. And also we wanted to make sure that when we introduce the bacteria into the cornea, into the model, these were not washed off away during the experimental procedure. So I'm very happy to say that we were successful, and we did establish our model, and we are very happy with it. And we also investigated the actual effect of bacteria in this ex vivo model, the effect of genipin. Unfortunately, I cannot show you my real data because we are in the process of publishing this data. However, we still have a long way to go, but we are working hard to get there. And I would like to thank my collaborators at the Universidad Nacional de Colombia, my group in Cardiff, the Structural Biophysics Research Group, my collaborator, Dr. Pedro Castro, the European Commission for awarding me with my Marie Curie Fellowship and allowing me to carry these investigations, and of course, all of you for your attention.
Fantastic. Thank you. Thank you very much, It was very
You're
interesting
welcome.
talk. So does anyone have any questions they would like to ask? I've got the chat open, so if anyone wants to type the questions, or you can alternatively ask away. I think there's a little bit of a delay with the Facebook, so we might get some questions coming through there. I'm not sure. I will check. In the meantime, I had one question. So it looks really promising, that the treatment. Have you seen? Basically, I wanted to ask: does the treatment, apart from killing the bacteria, do you know if it also helps repair the damage already done by the bacteria, or is it something that? So is there a point of no return? Like, do you need to treat it? So basically, when does it become like? Is there a point of unrecoverable?
I cannot answer this yet, but all I can say
Okay.
is at the moment, so we had our in vitro model. We have induced the infection. We allowed some time for the infection to be established. Then we treated our corneas with with the drug and without the drug. And then we compare the bacterial growth. So we can see a huge difference, statistically different difference in the bacterial growth between the treated and the untreated corneas. However, we need to do more experiments to see whether this effect is permanent, whether it has, like, as you said, like it's just a temporary effect. Yeah, I still don't know the answers to these questions.
Thank you. Thank you. So does anyone else have any questions they'd like to just shoot our way?
Yeah, I've got a quick question. So first of all, thank you very much for a fantastic talk. It was really interesting. So you mentioned near the start that you have this sort of evolution sort of separating the eye morphology of all these different animals. And
yes,
it struck me, working on spiders and other invertebrates, that there are many examples where you have convergent evolution, where things like squid, for example, have these incredibly complex eye systems. And I was just wondering if similar conditions are seen in those species at all, and if so, whether this could be used as a zoological remedy as well.
Wow, that's very interesting question. To be honest, so far we examined shark cornea, we examined the eagle, the dog, the sheep, the chick cornea, but I don't know anything about the spider cornea. But if you want, when I come back to Cardiff, you can give me some samples and we can carry some studies.
I would love to give you some spider.
But thank you. But yes, basically, I didn't have time to go through those changes, but just to give you a brief overview. So basically, you can see that the collagen organization, it changes massively. It becomes more complex, but also with this complexity, it also offers the cornea better power to be for better visual acuity. And also, it also depends on the refractive power, so the difference between the refractive power of the cornea and the environment. So yes, it will be nice to see about the spider as well.